Great. Thank you all so much for having us, and thanks, Chip, for that introduction and for the orientation to the app itself. Hi, I'm Tracy Smart. I just waved at you and I don't have a camera on, so I don't know why I did that. But I'm going to start off talking this today, covering the first part of the presentation. I'll hand it off to Julie. I will pop back on towards the end. Wally is out enjoying some spring break time, so maybe he'll share some pictures of the SSC meeting in a couple weeks of his time in the mountains, but I'll cover his slides towards the end. So with that, we really need to start out with acknowledgements on here. If you have met any of us in person, you might be thinking, 1972, either those three are aging extremely well, or they have not been around for all of this stuff. And so we really need to acknowledge all of the survey and vessel personnel, agency personnel, as well as council, science center, grants personnel that have been involved in these programs from day one through the present day, that have supported, that have conducted the business when we have not been here. And we are simply stewards of these programs right now. They started well before us, and our plans and our hopes are to care for them, help them grow, so that they can serve other scientists and managers well after we are done. So we have been told these stories and enjoyed them ourselves, and enjoyed hearing about how these events and these decisions were made and changes were made in the past, and wanted to share those stories with others. And so we will try to do that today. All right, so I suppose starting back at the beginning. So in 1972, South Carolina Department of Natural Resources, before it was quite that brand, as well as the Marine Resources Division, opened its campus in Charleston out on James Island at the former site of Fort Johnson, opened in 1972, and MARMAP was one of its first programs. And if folks have been around for some of the assessments recently, some of the updates to the SSC or to Snapper Group or AP, you may have heard of the name MARMAP as part of our survey work, and that was one of the original programs out here. So we might as well start with what that means and what it stands for and what was it trying to do. So it stands for Marine Resources Monitoring, Assessment, and Prediction Program. And more specifically, its goals were to provide information on offshore species of interest to the region and of interest to management and economics in the region, such as snapper grouper. And by doing fishery independent surveys for these species, meaning that we were working under scientific designs rather than looking at the fishery dependent side. So not doing, intercept surveys or going out with commercial boats, but rather working on research boats, providing those data to folks doing assessments or making management decisions, such as the Science Center or the South Atlantic Fisheries Management Council, once the council actually existed in 1976. So we actually even predated them. And then prediction. The idea being that the hope would be that the data collected by this program or similar programs would be able to help inform what was coming around in future years, and so that we could plan and make reasonable decisions about managing our fisheries. So for example, if we were out there sampling young fish, would that be able to predict old fish a couple of years down the road, or would those young fish predict things like fish coming into the fishery in a couple of years? So MARMAP sort of took that to heart with the first grant, and in 1972 and 73, this started with ichthyoplankton and zooplankton surveys. So ichthyoplankton is mostly fish eggs and larvae, and zooplankton are things like copepods that fish would be likely to eat. So those were conducted from northern Florida through central North Carolina. along a variety of transects along the coast that you can see in that very original map from one of the reports in '73, and done by small mesh nets, either at the surface, such as the Newston net, or bongo nets through the water column. And that was one of our original research vessels owned by the state, the RV Dolphin. And so that would give an idea of the spawning productivity in a given year, as well as potential food resources for those very young fish before they entered their first winter. That sort of model of surveying was also adopted in other areas for other MARMAP programs, such as in the Northeast Fishery Science Center, which worked from Cape Hatteras to Cape Sable. They ran ichthyoplanktons from 1977 all the way until 1988. All right, so you know, part of developing these new programs was also thinking about whether or not they were reaching their goals. And those ichthyoplankton surveys were in fact encountering snapper grouper eggs and larvae, so that was good news. And they were doing so through a fishery independent survey. However, there was still some big unknowns on whether or not it was going to be data that would be useful for assessments and then ultimately for predicting future conditions for fisheries. So the next thing that was added to the programs in 1976 through 78 was groundfish and shellfish trawls, which use very similar to something like a groundfish trawl in the Northeast, as well as some of our shrimp industries in the Southeast and the Gulf of, sorry, in the Gulf. And you can see some examples of those that were run off the RV Dolphin as well. And those were meant to get species that would be either subadults or adult fish that we could relate back to what the ichthyoplankton surveys were seeing. But it didn't quite work out as the hopes were. I used to work up in Alaska, and we had a similar sort of system where we had ichthyoplankton surveys and relating them to things like juvenile fish surveys as well as the adult fish surveys. And in these high latitude systems, like in Alaska, the Northeast, for things like pollock and cod, you have very specific spawning and growth seasons for fish where you have very cold winters. And so those spawning occurrences kind of happen in very distinct pulses in the spring once temperatures warm up a little bit. For example, in walleye pollock, you might get spawning really specifically, mostly in May of a given year. And so eggs and larvae would be present in that area. And if you had an ichthyoplankton survey the way MARMAP did, and you sampled through that peak with your survey, you would get a really good view of what was being produced in a given year. And you could relate that to a couple years later with your other surveys. However, in the Southeast, we have very mild winters, we have really warm summers, and we have fish that have very, very long spawning seasons. Species like vermilion snapper might actually start spawning in May. They might spawn again in July. They might spawn again in September. And so if you have an ichthyoplankton survey that's only going out once a year, you're only getting a little snippet of the actual productivity of eggs and larvae in a given year. And so that information just doesn't connect as well as you would hope. And so the ichthyoplankton surveys were not quite as effective at predicting conditions as the hopes were originally. And additionally, at that same time in the 1980s, you know, more and more folks were really realizing how much of the bottom habitat that those subadult and adult trawls were disturbing as we were trying to estimate abundances and biomass of fish through the sampling being conducted by the MARMAP efforts. And you can see some examples of some of that live and hard bottom being brought up, covered by some of those soft corals, sponges, algae. And when you're trying to sample something like snapper grouper that are associated and are reliant on those bottom habitats disturbing or removing them is not the best approach. And to the point where in 1989, in the very first amendment to the Snapper Grouper Fisheries Management Plan, the South Atlantic Fisheries Management Council prohibited roller rig trawls between Cape Canaveral and Florida and Cape Hatteras, North Carolina. And so MARMAP really needed to address these concerns and kind of think in a conservation way and move away from destructive gear. So in terms of assessment, we needed to reconfigure what we were doing. So one of the things that happened about the same time is trawling efforts shoaled and moved and became the SEAMAP Coastal Trawl Survey. SEAMAP stood for Southeast Area Monitoring and Assessment Program, South Atlantic, so same elements and similar goals. And trawling itself can be really useful in places like soft sediment or flat pavement bottom types where the disturbance to the bottom is much, much less compared to hard bottom areas. So from 86 to 89, the program started net trials and developing techniques and survey coverage, and ultimately became the modern day SEAMAP Coastal Trawl Survey that monitors things like drums, bluefish, mackerels, shrimp, rays, and sharks, and sea turtles in our coastal areas from Florida to North Carolina. Now, one of the things that happened recently in this survey is we retired our long-term trawler, the Lady Lisa, and welcomed the RV Lady Lillian. And part of that was moving from a system that used 2 trawls nets on the outriggers to a single stern trawl net. And so we've been doing work on estimating the geometry by putting sensors on the nets to actually measure the height, the width, as well as the area swept of our nets, looking at our catch lengths of our fish that we collect to make sure that we're catching fish of about the same size or the same, and the nets are selecting the same fish from those nearshore areas, as well as estimating the same composition from these habitats. And part of that transition too is moving from that double net rig to the single net rig. And so looking at those ideas of whether or not we can convert from 2 outrigger nets to 1 outrigger net, and then from 1 outrigger net to 1 stern net, and then whether or not we need to account for anything, including things like a multiplier for the ship's shadow, et cetera, et cetera, et cetera. So we recently just published some of that work. It came out actually earlier this week, late last week. So, and with that, I'm going to pass over control to Julie, and she's going to tell you about what happened in the offshore area.
Right, so moving back into that offshore area, we can't use trawls anymore. So what are we going to do? And in 1988 to 89, MARMAP started testing several different styles and shapes of fish traps, pitting them head-to-head: blackfish traps, chevron traps, and that Florida Antillean Z-trap. And anybody who's familiar with these long-term surveys knows which one won. That's the chevron trap, which has been our standard gear of choice since the year 1990. And so we have set and retrieved thousands of these traps over that time period. Our targets using chevron traps are those mid and outer shelf areas, generally hard bottom, and the traps themselves are retaining shallow water groupers, snappers, triggerfish, porgies, and black sea bass. Since those first deployments in 1990, we have set over 31,000 chevron traps and processed over 340,000 fish for age and reproduction. The program has contributed to over 65 student theses and over 140 peer-reviewed publications from the survey itself. The sampling universe, of course, relies on hard bottom. And as you might be able to see from this map, we had a pretty good idea and pretty good communication with others that were out on the water about where that hard bottom might be along the South Carolina coast, but a little bit more sketchy about where we might be able to find some of those habitats and those species that we're targeting in the outer reaches of our survey. The MARMAP survey has always tried to serve from Cape Hatteras to essentially Cape Canaveral or even further south. And so again, you know, a lot of information about our immediate coast, but maybe a little bit less in those outer reaches. And especially difficult too, because of the technologies that were available at the time. We, of course, have always been a product of our time, including the ways that data is collected and recorded at sea, including, of course, using paper data sheets, paper fish finders, and Loran locations to figure out where we're going, paper data sheets, and hand design of dissection selection, and finally, writing by hand every single envelope and container for every single sample that might be coming off of a fish. These days, things are actually quite a bit better, much more technology involved, making our lives a little bit easier at sea, including digital capture of digital information from the bridge, like where we are and what the depth is, as well as digital capture of electronics of measurements, species identification, which allows for us to print out directly the labels and printing onto our tiny tissue text using a printer instead of having to handwrite them. As always, this program has really been focused on providing the best products for research and management. And when the stock assessment process moved into the SEDAR format, the first one that was finished in 2002, we were there for South Atlantic red porgy. Since that time, we have contributed to over 30 stock assessments in the last 25 years, providing information to most of them, such as indices of abundance, total numbers of fish that were available, length frequency distributions over that time, how big were the fish that we were encountering, age frequency distributions, and reproductive information to each one of those. And again, over 35 CDARs we've provided all that information to since the year 2000. About halfway through that time period, we've always known that this is a really big area and a really big place to try to adequately sample over the course of a summer using just a single boat, leaving from a single point right in the middle of that region. And so in from 2009 to 2010, we were able to include not only an additional funding source, but also a federal partner in the SEFIS program to create one unified Southeast Reef Fish Survey. Once that Southeast Reef Fish Survey was sort of finalized in place, we were able to essentially double our total numbers of traps and really intensify our sampling, especially in those outer regions. You can see how many more traps we're able to put in Georgia and Florida, as well as up off of North Carolina as well. So this has really improved the density and the coverage of the survey, especially in those outer regions. One question that began to be asked was, well, what happened to those earlier data? Were those still useful? And we did analyze this over the last couple of years and found that the indices that we produced for most species are still relevant and relatively right on when we compare a low density sampling to a higher density sampling. But for species such as red snapper that are more contained or centered in the southern area of the range, we really improved our ability to see changes in their populations. Now, we've been talking throughout about the need for that hard bottom habitat. And of course, we've always been really, really interested in seeing that habitat too, and making sure that we're really going to the places that we think we are. And so this has been an interest within the program over the entire period. Of course, the technology at some times was maybe leaving a little bit to be desired. You could kind of see that there might be something down there. But these days, we are actually able to see quite a bit more with the miniaturization and just improvements overall to our camera systems. Today, every single chevron trap that is deployed has riding on the top of it 2 GoPros that are pointing outward in each direction and able to record everything that's around that trap at all times throughout the entire deployment. So we're able to see the fall to the bottom, what that habitat might look like, as well as any species that do go in the trap and species that might not go inside the trap at all. Our NOAA partners are reading each and every one of those videos for the abundance of various species on every single one. And what we've found over the last 15 years of doing this procedure is that we have complementary indices. These indices are showing similar patterns for species that do go into traps. However, the fantastic addition that we have by including these video cameras is that we can now produce indices of abundance for species that are trap wary and that don't like to go inside traps. Both these example species, greater amberjack and mutton snapper, are very, very rare inside traps, but pretty abundant outside traps. Another thing that's been great about having these additional scientists on board and as collaborators is that we get to think about, ask, and answer a variety of new questions, such as, do these traps ever saturate? Are there species that are, is an hour and a half enough time? Is it too long, etc.? What's the area that these traps might be pulling from? How far away can we expect to see fish coming to the toward the traps themselves? And of course, along the way, we have also engaged in a wide variety of extra complementary and innovative projects. One of the great things about being on the water and being able to put 1,500 traps into the water a year is that you can then ask and answer a wide variety of additional questions using a lot of the same gear. So a few examples of our most recent questions that we've been asking and answering are looking at the size selectivity of our traps. Do chevron traps have a specific bias or size selectivity that might be different from some other gears that could be out there? So using stereo cameras, which is 2 cameras that are looking at the same thing so that you can get a measurement of the items in the frame, as well as an innovative gear that the FWC invented called a repetitive time drop hooked gear. We also have been looking at a variety of novel technologies for investigating sort of life history and ecology of the species that we're engaged with, including using infrared light for aging and sex determination. This technology is incredible because it is fast and inexpensive. Each sample to age a fish using this technology is about 30 seconds versus the hours of time and the amount of money that it takes to prepare a single otolith for visual identification and aging. In addition, we're trying out isotope analysis of the eye lenses of fish to get a history of individuals' ecology over their entire lifetime. And finally, this same grant includes looking at the stomach contents and diets of certain species, including red snapper and groupers, and black sea bass using DNA barcoding. So a lot of the diets of these fish, are unidentifiable, but using DNA, we can figure out the species related that were inside the stomachs of these fish. The survey itself has always had these boundaries of Cape Hatteras and sort of Cape Canaveral, or now extending down to St. Lucie in Florida. But one of the questions that has been asked recently is whether we can expand all the way out to the edges of the South Atlantic Council boundaries, right? And that is the North Carolina, Virginia border, so up and around Cape Hatteras. And in 2024, the NOAA ship Pisces was able to spend about 12 days up there, setting 84 reconnaissance chevron traps, as well as doing quite a bit of mapping to look for some of these hard bottom habitats throughout the region. The working farther south than Port St. Lucie is quite a challenge, and we've been thinking a lot about how to accomplish this because of a variety of different challenges that that occur in the region, including that the shelf is incredibly narrow with very high currents. Many of the target species are trap avoidant, and we know it. And most of the area is covered by a special permit that's required in order to set anything there, as well as the sensitive corals that live in this area. Nevertheless, in this summer, we do plan to try a new sampling design throughout the region. This will be a collaboration between the Florida Fish and Wildlife, NOAA, and our program, setting a few different styles of camera pods for a few different reasons. First of all, because we know that a lot of the targets are going to be trap avoidant. Also, this reduces our total footprint, so the amount of space that the size of the item that might be falling near a coral. We'll be using stereo cameras again to get measurements of these fish, as well as side scan sonar, and all of this to get species composition, total counts, lengths, and habitat information throughout the South Florida region. One final additional project that I wanted to mention is one that we're trying out for the first time, which is looking at comparing the artificial and natural reefs off of the South Carolina coast using our chevron traps. We're doing this both in the winter time and the summertime. And one of the reasons why chevron trap survey has been traditionally out of the water during winter is because of the migration of right whales through the region. However, recently, there has been new inventions to provide for buoys that are held down next to the traps on the bottom until they're contacted by using a pinger, and then that float will rocket right up to the top, and the vessel can come around and scoop it right up.
Right, and I'm going to tag back in. So we've moved from shallow water into the mid and towards the shelf edge, and now I'm going to take us to the shelf edge and a little bit off. So MARMAP also had a history of working in deeper water at the shelf edge into the very deepest area for tilefish grounds, up to about 600 meters or so. And that started back in the early 80s. So from 1982 to 1986, they utilized Cali poles, which seemed to create a giant mess on the deck of fish everywhere, hooks everywhere, as well as these poles everywhere. But it was really effective for high relief, hard bottom areas, in particular those that were expected to, host snowy groupers in particular, but also blue line tilefish. Occasionally you'd get a golden tilefish on those as well. And these were also, mile or so long, so they could sample actually a fair amount of area. If you recognize that little red guy too, that's a black belly rosefish. Super fun little scorpion fish in that area too of Deepwater. But the Cali pole was a little bit logistically challenging. So in the 90s, we changed gears literally a bit to focus on 2 different types of habitats with 2 gears that were a little bit more appropriate for each one. So our bottom long line was for soft bottom habitats that would be more likely to host golden tilefish. And these were mile-long cable long lines that were baited with squid. And these were primarily deployed from either our RV Palmetto or the Lady Lisa. In the more recent years, you can see some of the folks that are baiting up and getting ready to deploy the long bottom long line there in the early 90s. And then for our high relief hard bottom habitat, the short bottom long line was used, and this was a 75-foot rope ground line. So fewer hooks, again, baited with squid, and very similar to the chevron traps. These would soak on the bottom for about 90 minutes or so before they would be retrieved. And so the short bottom long line would primarily be areas that would be likely to house snowy grouper again, as well as maybe blue line tilefish. Jacks were very common on it as well. However, the challenge with these Deepwater surveys for fishery independent group was that the run times and distances to get to these areas, especially off of South Carolina, Georgia, 60, 80 miles to the nearest areas for sampling, a little bit less off of North Carolina, southern, central Florida. But to cover the whole region, which was challenging enough for traps, was extremely challenging for deeper water work. And so that was always one of the critiques that was received, was that it just wasn't sort of comprehensive enough. And so these 2 gears really became less used and ultimately disused in either the 2010s, and then finally the short bottom long line, its last year of sampling was in 2022. Following the start of the South Atlantic Deepwater Longline Survey, which is a collaboration with NOAA, as well as SCDNR, Wally Bubley in particular, and Kevin Spanik here work with the SADL survey. And this is a little bit different. It is still sort of fishery independent techniques, but it's cooperative research using commercial fishing vessels, working very closely with the captains and crews of those vessels to conduct the surveying work, using NOAA observers on board to work up the fish. We work up the life history samples here in-house, and the developing the indices of abundance is sort of to be determined. I know NC State as well as the Science Center are currently working on that. And folks are very interested, and they've reached their 5th year of sampling. So this is sort of the latest and greatest and where Deepwater work in the area is going next. So you will very likely see SADL survey data very soon for species like snowy grouper, blue line tilefish, golden tilefish, and they do capture things like large red snapper in that deeper water. So, but I do want to note, this is a long time period, 50+ years. Some things have certainly changed over time. Our techniques have changed, our kind of attitudes on what we're doing and how we're doing it. Our awareness of sun safety, as well as use of sunblock and sunscreens and protective clothing have changed. But honestly, some things have really not changed over time. Our folks still really enjoy their jobs. They enjoy providing things that are extremely useful to science itself, as well as the management personnel, and doing work that is, you know, meaningful to folks, that you can explain to your parents about why you spent how many years in school for this. And so with that, we'd just like to thank you all for listening. And yeah, if you have any questions, we're happy to answer. Thank you.
Thank you for a great presentation. Let me take back control. And then once again, just a quick reminder on how to ask a question or raise your hand. I'm just going to go through one of them. You can type it into the question box, or if you see a question mark on your screen, just type a question in there. If you'd like to ask a question, you can also click on the hand icon. Here, it's shown as green. If your hand is red, that means your hand is raised. And then we'll unmute you. Make sure your microphone is green when it's ready to speak, when you're ready to speak. And we will let you ask Tracy and Julie any questions you might have. And so with that, yeah, let's move into the question and answer. And so I'll start off with one, since I'm not seeing any hands raised yet or any questions in the question log. Maybe it'll be easy, maybe not. You probably weren't expecting this one, but two questions, or I guess a single question with two parts. What is the most referenced paper, and what do you think is the best paper that's come out? Since you guys have had 140 papers, giving us a short primer on which one we should be reading, I think would be nice.
Collins 1990, which is the dimensions of the chevron trap and the comparison amongst the traps, and that it had the broadest community composition and size range of all the species of all the 3 traps tested by MARMAP. Is that the one?
That's definitely the most cited paper.
It's the quintessential trap paper. So I'm gonna go with that. I'm gonna vote that one.
All right. And Judd, I see your hand is raised. Go ahead.
Can you hear me, Joe?
Yep.
Yeah, thanks, Tracy and Julie. Great presentation. Such invaluable data for stock assessments and things going forward. And it's cool to see just the use of different technologies and as they're advancing and how you use them. And so my question, I guess, is as you're starting to see new technologies emerge, things like AI. What are, like, let's say, infinite funding and resources? What would be some of your I don't know, the highest kind of level ideas or things you'd like to explore or improve upon on the survey, either brand new or extrapolating on what you've already got?
Yeah, I mean, I think specifically AI related, there's been high hopes for, I don't know, at least 15 years about the utility of AI with those video sections of the survey, and especially if we're using stereo cameras to get some measurements. I really love this technology as an additional gear. I don't think that it stands on its own. But one of the biggest hurdles with any video survey is just the amount of time that it does take to analyze those videos and get all the way to measurements. And so, if we really can capitalize on the improvements of AI for that video reading, I think that could be a really great place to really improve efficiency. So far, it's not quite there yet as far as what I've seen, but I think, infinite resources, it would be there quickly.
Yeah, I think the other complement would probably be in the bottom mapping and characterizing of habitat, because it is such a large area, wide, shelf, and our universe for the traps is points, and you know, mapping is done in swaths and somebody has to physically go in and characterize those. So I think that could be a huge help. From the trawl survey perspective, even that would be helpful because we know there are some areas that are true sand versus pavement. And, you know, we get, for example, you know, our true soft sediment sort of associated demersal fish like drums, cyanids, rays on the soft sediment, but we get black sea bass young of the year on those hard bottom pavement areas. And if we knew better where they were, you know, it could really change how we might approach things or how we might pull out data specific to those stations if we had even better, more detailed mapping and sub sand, what we can see with a camera, say, for those near shore areas.
And yeah, I was thinking about the mapping that's come up in a couple recent reviews of some projects as a critical resource or a critical source of information that we need here in the South Atlantic. See, Jimmy Hull has his hand raised. Jimmy, it's showing you as muted right now. Jimmy, you're still muted if you're speaking. Okay, Jimmy, we're not hearing you. It might be good for you to type your question into the question box if possible. So my next question I have for you all, as we wait for Jimmy's question to come in, you showed the near infrared spectroscopy, and I was just curious about that process that you're using. You know, how do you validate ages with that process? Do you have to go through and it's a known age fish? Just curious if you could expand upon that a little bit.
I was actually just looking to see if Claire was on the webinar. She's not, she's hiding from it. Yeah, we have a graduate student right now that's working on that with Wally. We have a near infrared. This is why I let Claire do this, because then I don't have to say the word. We have a NIRS machine here. How much detail are you looking for here? Are you looking for like excitement of compounds? Yes. Yeah. So there are traditionally aged fish, and the other otolith that was not mounted in section for the traditional aging is used as the paired experiment for NIRS aging, where the light is shown through it and the compounds excite and do the thing and like emit, and it's recorded. And I should probably know this way better than I do. And so that could, so you're allowed to do that one-to-one comparison.
Okay. And Jimmy is back on now. Jimmy, if you want to go ahead and ask your question. Still showing you as muted, Jimmy. Jimmy, if you click on your audio, click no audio, then back to your computer audio, sometimes that will fix hang-ups that you might be having. Nope, that didn't work. All right, we'll continue to work on it for Jimmy. So you also presented on DNA barcoding, and I was curious if with that DNA barcoding, is it able to see like numbers of individuals, or is it more of a presence of or absence of species?
So there's a few different processes that can be used for DNA barcoding in diet specifically. The one that we're using right now is actually to identify unidentifiable organisms or pieces of organisms inside the gut. So what happens is this is often used primarily for fish because they get digested enough that you can't tell what they are, but they still have plenty of DNA left in order to identify them using DNA. And so those sort of chunks of half digested fish tissue are individually sampled. The DNA is extracted, amplified, and then identified. So those are able to be tied back to individual prey items within the diet of an individual species predator that you're looking at, like a red snapper.
Okay.
Should make Jenny to answer that.
And let's see, no other questions quite yet. So I will keep asking away till somebody else will make me be quiet. The next thought I had was, where could we, you've mentioned 140 papers. Obviously, that will be a challenge to track down. Is there a place where we can find all your papers? Are they? Is there a list or links or something like that available?
We actually have an EndNote or some sort of library here in-house. Our website is undergoing a revamp right now, so it's not on there, but I'm sure there's a way to distribute that if folks are interested.
I got the 140 number by literally counting the PDFs in our server, so we have copies of all of them.
Well, I appreciate the effort. That doesn't sound like a lot of fun. Hallie has her hand raised, so I'll go to her. Go ahead, Hallie.
This is not a scientific question at all, but what's the weirdest thing that you've pulled up or encountered?
Oh, that's a good question. Like biologically weird or just in general?
I think in general would be more interesting.
I mean, it depends. So my favorite thing I've seen recently was on the video. If you were on the Cedar 95 call the other day and watched Nate Batchelor's video index for cobia presentation, there's a clip of a tiger shark wearing a shark sucker as an eyebrow, and that I enjoyed thoroughly. Sea turtles, probably shouldn't talk about sea turtles ramming into the traps, but they do regularly. At nighttime, if we're anchored and the lights are all on the vessels, sailfish will come up to the boat, and that is amazing to see them super close up. They're really curious. We thought we discovered a brand new shipwreck once. Turns out it wasn't. People knew about it. It was on actually a chart. It just wasn't on our chart. That was a little disappointing. I mean, it was an old shipwreck. It wasn't like new. I don't know. You got anything?
Regular flybys, especially off of maybe North Carolina and Florida. We get regular, flybys or visits, sort of close encounters with naval vessels and things like that sometimes.
Rocket launches. My favorite was one of our D-hookers was on one of the traps when it went down, it came back. That was a great day because usually they don't come back. I don't know.
Those were all
I mean,
great answers.
So along those same lines, what are some of the biggest changes you've seen in the surveys over time, whether it's the trawl survey or the trap survey?
the red algae off of southern North Carolina 2 years ago, I want to say it was 2024. I don't know if folks on the call remember that. The shrimpers were definitely complaining about, it was more of a filamentous red. I never figured out what species it was, but it was really common in our trawl catches, and then we saw it offshore a fair amount that summer too. Last year, it was super fun to see the upwelling and see it move along the coast. We saw it in Florida early in like May and saw it progressing up the coast as we kept sampling, and we hadn't seen a good upwelling like that for several years. And it was fun to see it in you know, live in action in real time. And also, I really enjoy it when I get to see vermilion and tomtate start picking on lionfish, which they do regularly now.
So you can see that in the videos where they're pestering the fish?
Yep.
The lionfish?
Yep.
They're not scared of the big bully anymore.
Nope.
All right. Not seeing any other questions. And we will give you back 30 minutes. We greatly appreciate the presentation today. And, you know, I think this can be a source presentation. So when people are asking why are certain surveys set up the way that they are, I think you guys did a great job covering the history of South Carolina DNR in your fishery independent surveys, and also some directions of the future on what's going to be happening. So we greatly appreciate all the information. And yeah, do you all have anything that you'd like to finish up with?
No, just thank you. We enjoyed pulling old pictures, so.
Yeah, it was great to see how this thing has operated for so long, and you guys are going to continue to work with it going into the future. So with that, I will thank everybody for coming today, and I apologize for moving the normal date and time, but we had the SSC meeting double booked for us next week. So this will be on our website later this week, and if you want to share it with others, that we're expecting it to be next week.
Oh,
So
I will add a plug that we'll be presenting data at the SSC meeting next week.
Yeah, data from all 3 surveys, right?
Maybe not SADL just yet, but yeah, at least 2.
Okay, excellent. Looking forward to it.